Okay, are there any questions?
I had a question on lab 6 for number 7. It says, when observing a zone of inhibition around a disk, how would you determine whether the antibiotic cause of the zone was bactericidal or bacteriostatic?
I haven't finished grading that lab, I think. Let me check on that. Uh, there is a way to tell. In fact, there's two ways to tell. And I guess I'll just go ahead and state one of them. You can't tell by looking, okay? But you can tell by streaking the zone of inhibition. If you streak the zone of inhibition on another plate that doesn't have any antibiotics on it, if the zone of inhibition killed the cells and was bacteriostatic, no, bactericidal, killed the cells, then you'll get no growth on the other plate. However, if you streak the zone of inhibition and it's a bacteriostatic antibiotic, you will get streaking on the plate that has no antibiotics. Okay?
Okay, that makes sense.
And you had another question which I never answered earlier because it was taking up too much of the lecture time. I was trying to answer that now, and it was on the infectious disease project. And you wanted to know specifically about talking about the proteins that cause necrotizing fasciitis, what is it, the toxins?
Yeah, it's the M protein and I think it's toxin A. I can't remember.
Let me show you my screen. I don't think I'm doing that yet. So if you come down to the grading, this will be a little difficult to read because it's in the web page, but I'll blow it up. Pathology. Describe the pathogenic effect on cells and tissues. Let's see if I can scroll down. Not easily. Nope. Let me shrink it. Describe the pathogenic effect on cells, tissues, and organ systems. What tissues, organs, cells does the pathogen affect? What damage does the pathogen inflict? So when talking about the damage the pathogen inflict, you would have to talk about the toxin causes some of the damage, and how is it inflicted by the production of toxin. And so you should talk about that. Oh, in this whole pathology, you should have at least one paragraph. And so talking about the damage the toxin does, you should discuss that in a sentence or two. Okay?
Okay,
Any question
so
about that?
if like exotoxin B is like a protease, would I just say like how it lyses red blood cells and stuff like that?
Yeah, just talk about it in a sentence or two. Okay? So
Okay,
you
that
do
sounds
want to
good.
talk about that in the pathology. My point is you can't just ignore it. You do need to discuss it because it is a toxin produced by the pathogen. And I think that would be it. All right. Sorry that took so long, but I wanted to specifically point out to you what I was talking and show it to you. And I had to switch to different computer to do that. And and it was all a mess. And so I just said, we've taken up enough time of the lesson and we're behind schedule, and now we're further behind. So, all right, let's talk about today's lab. I think I answered the earlier question for you. Is that right? Did you have another question?
Just a short one. So for the pathology part 4, how do we go and find like the time sequence of disease on the internet? Because I was having trouble looking for that.
Let me see if I still have that here. I don't have it here anymore. Oops, I just shut that too. That wasn't right. Just if you don't find it on Google, go to Wikipedia, and don't put Wikipedia as a reference, but you can go down in Wikipedia's references and give a more concrete reference. But you should be able to find someplace online. What was it? The time sequence of how the disease acts or something? What was your question exactly?
Like the length of incubation and how long it takes until it causes illness.
Yeah, that you should be able to look up. Look up the disease and what's the incubation period from when the person first became infected to when they first had signs and symptoms. That should be stated somewhere. It should be discussed under the disease. All right, any other questions? If not, today you'll begin work on the unknown work, and I'll try and email you your first results either after the lab and maybe even hopefully during the lab. Okay, when you guys are working, maybe I'll email it out. Today we're discussing lab 7, throat and urine cultures. So let's go to lab 7. There is some reading in the textbook, not too much, but please read that on the different tests. There's some video clips. There is no virtual lab because the virtual lab uses a program which is no longer supported, and so it won't run. Upon successful completion of the lab, you should be able to explain how to properly collect throat and urine specimens for routine culture. You should be able to list common bacterial pathogens isolated from the throat and urine cultures. You should be able to know the culture media used to cultivate these pathogens. You should be able to recognize which tests are used to identify throat and urine pathogens. And then you should be able to understand the principles and how to interpret the following tests: the catalase test, the coagulase test, and the oxidase test. We will briefly talk about the XV or the 15 factor testing, but I'm not going to ask you any questions about that because it's a complex test and we don't really go into it in any great detail in the lab. So once a specimen is received in a lab, the specimen will be logged and then processed in a timely fashion. And it'll be cultured, inoculated, and incubated for a specific time, looking for human pathogens. Today, we're going to be talking about throat and urine specimens. Below is an overview of the culture media used to cultivate common bacterial pathogens isolated from the throat and urine cultures. So from the throat, we have two main media that we culture pathogens from. First is blood agar, and second is chocolate agar. The most common bacterial pathogen obtained from the human throat is Streptococcus pyogenes, which is grown on blood agar. Haemophilus influenzae is a human pathogen which is commonly seen in children, particularly infants and young children. And it does not grow on blood agar. You grow that on chocolate agar. For urine pathogens, we tend to use 2 of 3 culture media. So you always use 2, you don't use 1. We will first grow the urine culture on blood agar, and then either on MacConkey's agar or EMB agar. We'll talk about why we use MacConkey's agar or EMB agar, as well as blood agar in a little bit. Common bacterial pathogens we isolate from a urine culture are Streptococcus species, Enterococcus, and Staphylococcus, and all of these are gram positive, and they're all cocci also. But there are some gram negatives that come from urine cultures, and these include members of the family Enterobacteriaceae, specifically mentioning E. coli, as well as Pseudomonas. These are gram-negative bacilli. All right, first throat cultures. If you look in the throat, you see white patches on inflamed tonsils or white patches in the back of the throat. That is indicative of a bacterial infection. If the throat is just red and inflamed, that's probably a viral infection. But if there's white patches on a red throat, on the tonsils in the back of the throat, it's a bacterial infection, or most likely a bacterial infection. The most commonly isolated bacterial pathogen is Streptococcus pyogenes, which has a few other names, such as group A Streptococcus, and that's a term I don't use, okay? Or gas for group A Streptococcus. And the lab does use, I think it uses group A Streptococcus, but I like calling it specifically by the species name, Streptococcus pyogenes. So a viral infection and then a bacterial infection. Strep throat can be diagnosed, and that's strep throat is caused by Streptococcus pyogenes, can be diagnosed by a rapid test or a throat culture where you swab the back of the throat and the tonsils. You do not, when you're swabbing the back of the throat, you avoid the tongue and the sides of the mouth, meaning the gums and the forward part of the throat. Shown here. Patients who test positive on, and then you streak that on a blood agar plate, which isn't highlighted there, or should have that highlighted. Patients that test positive for Streptococcus pyogenes are then placed on antibiotic therapy. There are only 2 tests you need to do to show that a patient has a Streptococcus pyogenes infection. First of all, you grow the throat culture on blood agar, and then you look for small white colonies with complete zones of hemolysis, meaning beta hemolysis. And then you'd follow that with 2 biochemical tests that are definitive to isolating Streptococcus pyogenes. So a blood agar plate that has complete zone hemolysis. You then run the catalase test, and then the basis trace and susceptibility test. Streptococcus pyogenes, shown here, I'll try and blow that up a little, would be the small white colony inside the circle, and that clear region, which is actually pretty big, is showing you complete hemolysis of the blood agar. That would be a normal throat pathogen, which right here has no hemolysis, and then right there would be some alpha hemolysis. Streptococcus pyogenes is catalase negative. Most bacteria that can be grown in air break down hydrogen peroxide by using catalase. Meaning here we have hydrogen peroxide in the presence of catalase. We generate oxygen and water from the hydrogen peroxide. And this creates bubbling, the oxygen in the hydrogen peroxide. If the bacteria are positive for catalase, oxygen and bubbles are generated. But Streptococcus and Enterococcus species are negative for catalase production. Any questions there? So now we know that it is beta hemolysis on blood agar and catalase negative, but that only says that it's a Streptococcus species or an Enterococcus species. We then do the bacitracin test, and there we take the throat culture and then plate it on another blood agar plate, and then put a bacitracin disc on the plate. If we have a zone of inhibition, which is greater than or equal to, I don't see it here, greater than or equal to something like 12 or 16 millimeters, we say that the bacteria is sensitive to bacitracin. And if it's sensitive to bacitracin, then it is Streptococcus pyogenes, also called group A strep. If, on the other hand, the zone of inhibition is less than that, or there's no zone of inhibition, then it is not Streptococcus pyogenes. Okay, any other questions? So you do 2 tests to show that it's Streptococcus pyogenes. You do the catalase test, and Streptococcus pyogenes will be catalase negative. And then you do the bacitracin test, and it'll be bacitracin positive. Another organism that's clinically significant and isolated from the throat is Haemophilus influenzae. It's a gram-negative coccobacillus. Haemophilus influenzae is fastidious, and it will not grow on blood agar directly. Haemophilus influenzae cannot lyse the red blood cells and then get the nutrients from the red blood cells. But if we lyse the red blood cells by growing the Haemophilus influenzae on chocolate agar, Haemophilus influenzae will then grow because the nutrients in the red blood cell have been released, and then the Haemophilus influenzae will grow. Another thing you could do, and I'll just talk about it briefly, is you could streak Haemophilus influenzae this way on a blood agar plate. And then you take Staphylococcus aureus and streak it this way in a single streak down the blood agar plate. What happens is the Staphylococcus aureus lyses the red blood cells in this region here. And in this region here, let me blow that up a little bit more. In this region here, you can see Staphylococcus, not Staphylococcus, Haemophilus influenzae growing around the streak of Staphylococcus aureus. And that's because the Staphylococcus aureus has lysed the blood cells in this region, releasing the nutrients from the blood. And now Staphylococcus aureus can grow in this region. But you'll notice Staphylococcus aureus is not growing in this region of the plate. And that's because it's too far away from the Staphylococcus aureus. And that is actually showing you the V factor and the X factor, which I'm not gonna quiz you on, meaning I'm talking about it briefly, but we're not really gonna, you don't need to know it because I'm not gonna quiz you on it. Any question about that? All right, if not, let's move on to the urine cultures. Urine cultures are taken to diagnose a urinary tract infection or UTI. Bacteria can travel from the urethra up the urethra and into the bladder. This is primarily a problem in females because they have a shorter urethra than males. And so they're more likely to get a UTI. To collect a urine specimen, you have to clean the genital area prior to collection. And then when you're collecting the urine, you don't use the start of the urine or the end of the urine. You catch the, quote, clean midstream of the urine, the middle portion. This reduces the cultivation of transient organisms that are collected from the urethra. The urethra have a normal microbiota, and you'll get that in your collection, but we can reduce it if we take the urine midstream. A better way to get I guess it's shown here. A better way to get the urine specimen is to put a catheter into the bladder and then sample the urine in the bladder. However, this isn't the best way because as you're putting the catheter up the urethra, you can take bacteria from the urethra and then deposit it into the bladder. The best way to do it is actually suprapubic aspiration, where you put a needle into the bladder and you sterilize, or not sterilize, but degerm the skin and then put the needle into the bladder and then sample the bladder urine directly from the bladder. However, patients are not particularly happy about the suprapubic aspiration because they have to get a shot, and they're not particularly happy with the catheter because, well, you have to take the catheter up the urethra. So usually what we do is the clean catch midstream. And then we have to just realize that not all of the bacteria that grow may have actually been in the bladder. Some of it came from the urethra. Once a specimen is received in the lab, 0 or 0.001 mil or 1/1000th of a mil of the urine is placed onto a non-selective media such as blood agar using a special inoculating loop that only measures 0.001 milliliter. Let me show you that loop here. There it is there. And there's a penny to show you how little that is. Can you guys see that? It's a little dark there. It's a little better that way. Now let me shrink that. And that lost my place. All right, here we are. Let's blow that up a little bit. We should grow the urine specimen on a non-selective media such as blood agar. However, we should also take the urine and put it on a plate that grows gram-negative bacilli, such as MacConkey's or eosine methylene blue. Both of these plates are selective for gram-negatives because coliforms are the most common isolate of a human pathogen obtained from a urine specimen. The number one pathogen obtained from a urine specimen is actually E. coli. And coliforms grow on MacConkey's and eosine methylene blue, and then the gram positives will not grow on these plates. But there are some urine specimens which are gram positives, and so we have to grow it on a blood agar plate. Any question about any of that? The interpretation of a positive urine culture is multifaceted. Some factors that need to be considered are the type of collection. Is it a specimen, meaning we put the needle directly into the bladder, or is it a mid-clean catch specimen where the patient is voiding and then collects some urine midway through the urine stream? You have to take into consideration the gender of the patient. Males have less bacteria in their mid-collection stream than females. And then you also do the number of colonies present per the mil of urine and base your interpretation on that. The presence of polymicrobial growth, meaning multiple organisms isolated, may indicate poor collection technique. Okay, usually a patient has one or a few species causing an infection. And if you have polymicrobial growth, it's probably indicative of a poor collection technique. Once you get colonies growing on either macaque or blood agar plates or both, we then do a few tests to try and interpret the pathogens. We run the coagulase test, and the enzyme coagulase is used to differentiate Staphylococcus aureus from other staphylococcal species, which are coagulase negative. Coagulase contributes to the pathogenicity of Staphylococcus aureus by converting the blood plasma protein fibrinogen into fibrin, creating a clot. The clot is a problem with Staphylococcus aureus because the immune system will have a harder time getting through a clot. So we have fibrinogen, usually in plasma, and in the presence of coagulase, we get a clot, fibrin clot. What you do is you can have a slide test or a test tube test, and I'll show you the test tube test. And you'll get clumping in the slide test, and you'll get clotting in the test tube test, okay? Whenever there is coagulase. If there's no coagulase, there's no clumping and there's no clotting. All right, any questions about the coagulase test?
No, why do they not use the chocolate agar on that though?
Meaning, why do they use blood agar instead of chocolate agar?
Yeah.
Probably because most of the human pathogens are hemolytic, so they will grow on the blood agar plate.
Okay.
Okay, there's probably not many of the human pathogens which are not hemolytic, and then you would have to use chocolate agar. The most commonly isolated pathogen from routine urine cultures is E. coli, a family Enterobacteriaceae. This species, this family, will grow on blood agar, but you'll also get gram positives growing on blood agar. And so this is why we grow the urinary specimen on both MacConkey's or on EMB. It will grow the family Enterobacteriaceae and other gram negatives.
Does Pseudomonas grow on the blood agar?
Yes, it does. Blood agar is very rich. It will grow most things, not everything, but it will grow most things. Everything that'll grow on tryptic soy agar, at least as far as I know, grows on blood agar. And then things that won't grow on tryptic soy agar, like Streptococcus pyogenes, will grow on blood agar. There are some other gram negatives which can cause a urinary tract infection, and they are not members of the Bacteriaceae family. These include Pseudomonas aeruginosa of its own family, the Pseudomonaceae. How we can separate the Pseudomonas from the Enterobacteriaceae is that the Pseudomonas do not ferment the sugar glucose, and they are all aerobic. The family Enterobacteriaceae are anaerobes. They will grow with aerobes, but they're also anaerobes. And they all ferment the sugar glucose. Another quick way to separate the Pseudomonas from the Enterobacteriaceae are to look for the enzyme cytochrome oxidase C, which is involved in the electron transport chain, which I just talked about in the lecture. And this uses the oxidase test. And in the presence of cytochrome C oxidase test, the oxidase test turns blue or a purple blue. And Pseudomonas has cytochrome C oxidase, so it turns the oxidase test blue or positive. The Enterobacteriaceae do not have cytochrome C oxidase, so they will be negative in the oxidase test. All right, any questions about the urinary test? The growing urinary pathogens. I guess I'll word it that way. If not, there's a summary. You can read through that. And there's a few terms you need to know. You're not going to be performing any of the lab because everything we're doing is online this term. However, you are responsible for knowing how to perform the following tests. The catalase test, and we have a little video on that here. The coagulase test, and we have a little video on that. And then the oxidase test, and we have a little video on that. The virtual interactive laboratory tests don't run. And then answer the questions for the lab, but don't answer it in the lab module. Answer it in the lab exercise or the worksheet. And the worksheet has the links to the videos and then the questions. And then when you're done, submit the answers to the question. And I will be here until, about 8:20 to help you answer questions. Although I think I'm going to go to the bathroom. So if I'm not in the camera, you'll have to wait until I come back. And I will try and email you the first test results as well. Any questions about the lab?
When you guys had like in-person lab, were you really taking like urine samples and stuff?
We were not taking urine samples when we had in-person labs. However, we did have the students run the catalase test and the oxidase test. I'm not sure we ever did the coagulase test. Okay, but not on urine specimens. You would run these tests on your unknown bacteria. All right, if there's no further questions, go ahead and start working on the lab. You'll notice there's one question which is crossed out. That's 8C. You don't need to answer 8C, okay? Because it's not discussed in the lab. You have to look it up online. I just decided you don't need to know it. All right. Go ahead and get started. I'm going to stop the share and stop recording.